Hallo daar.
Yeah. Okay. Well, people are still coming in. You're late. You won't get lunch. Always those Dutchmen. We have a reversed order now. We start with task 5 and go back to task 1. Or we go back in time, if you want to say. We start with embryos and end with the gametes. Okay, so thank you very much for having for giving me the opportunity to give a very tiny section of what we have done in the work package together with people at INRA, Florence Guignot. We've been working on embryos. I will also tell a little bit about oocytes. And I want to, my contribution to it was tiny, but my contribution was on the osmotic events during cryopreservation. And with that, I mean changes of the cell volume or changes of the salinity, the concentration of solutes inside the cells and around the cells. And one example of that is what you see here, that we have cells that are going to be frozen in normal conventional freezing methods, where ice is formed outside the cells, and therefore the remaining unfrozen fraction that contains the cells will have an increasing concentration of solutes, and that draws out water from the cells, resulting in a shrinkage of the cells. And therefore the intracellular concentration of solutes will also increase. That's one example, but there are other examples. Even before freezing, if you add just the cryoprotectant, which we often call CPAs for short, for cryoprotective agent, if you do that, cells will first shrink very rapidly, they will lose a lot of water. At the same time, they will have an influx of cryoprotectant into the cells, and eventually this leads to a reswelling of the cells to regain the original volume. So these are osmotic events that must be understood and controlled. The opposite happens when we remove the cryoprotectants after thawing. We need to bring the cells back from a high concentration of cryoprotectant to a normal medium. And what happens then is the opposite. First we have an influx of water, very rapid rise in volume, followed by a decrease in volume to the normal starting volume of the cell. If we would do this in one step from 1.5 molar to 0, cells would swell too much and they would explode, literally explode. And that's why these kind of insights in what is going on can help us, because now we have a protocol in which we do it stepwise, and we can actually calculate how big these steps must be. So this way of looking at it, this understanding, but also if you make it quantitative in a mathematical model, it allows you to do this not only qualitatively, but more quantitatively and accurate and specific. And it allows us to look inside the cells. So that's miraculous, of course. If you look at this picture that you've seen before, we can see qualitatively that the cells have shrunken, but can we also understand what are the concentrations of solutes inside the cells? And we can by using this mathematic approach. To do that, we need only two differential equations. The one describes the flow of water across the membrane, and the other describes the flow of the cryoprotectant across the membrane. And these look like maybe complex, but it's not complex at all. What these formulae do is they simply do the same as if I would open a window here. I would get a flow of air, and this flow of air is equal to how far I have opened the window multiplied with the pressure difference of the air. So that's how simple these equations are. In other words, the flow is equal to the openness of the membrane, the permeability, multiplied with the pressure difference for water or the pressure difference for cryoprotectant. So they're really very simple equations. And with these equations, people in the past have used these for predicting the optimal cooling rate. As early as 1963, Peter Maser has done this, and he could introduce a fixed constant cooling rate of, let's say, 200 or 100 or 25 degrees per minute, and then calculate how well do the cells prevent intracellular ice formation. And then later this was done by Jun Liu and John Kreitzer's group, in which they also included the movement of cryoprotectant. So that's nice, but all was done with a constant cooling rate. And then later, in 2004, we have introduced a different approach in which we have said, no, we don't want to have a pre-assumed cooling rate. The only thing that we say in advance is that the cooling must be as fast as possible to prevent slow cooling damage, so-called slow cooling damage, but it must be slow enough to prevent fast cooling damage, such as intercellular ice formation. And for that we have derived these auxiliary equations with which we can then describe a freezing protocol, a freezing program. And you can see this is the decrease of temperature over time of your straw or whatever you have that you want to freeze. And you can see that it is going down in this fashion, and it depends on the cooling, on the concentration of cryoprotectant that you are using. So in some protocols you would need a sigmoidal freezing curve. So our model can predict a non-linear sigmoidal freezing curve, which is in fact in line with empirical observations and which is in line with fundamental cryobiology. Now we have also applied this kind of modeling to what happens during addition of cryoprotectant for vitrification of oocytes and embryos. This is done within IMAGE. And as I showed you earlier, cells first shrink, they lose a lot of water, and then they reswell again in the original protocol from 1985 from Greg Fahey and Bill Roll. This is what is going on. In the second step, you increase the cryoprotectant concentration once more, so they shrink again, and then you plunge them into liquid nitrogen. But as I told you, we can look inside the cells. We can look inside the cells with this mathematical approach. So we can tell you what is the influx of cryoprotectant inside the cells. For instance, in terms of the volume of cryoprotectant, the amount. But we can also calculate it in terms of the molar concentration, so mole per liter, or mole per kilogram water, or osmoles of the total solutes. Now this is very important because this will tell you whether there will be a chance of intercellular ice formation. So this is very critical. Now, I collaborated with a group in Ghent. They used this kind of vitrification of oocytes, and they were the first to be successful with the vitrification of immature oocytes. And they did that with using this very short protocol. Which is nothing new because other people at Utrecht had done that earlier in 2007 and 2009. But the other group was not successful, that was the difference. But that can be explained. Anyhow, it was the same short protocol. Nothing new, but of course these people in Ghent were worried a little bit because if it is so short, how can we be sure that enough cryoprotectant has entered the cells? So we turned to using this kind of modeling, and I could show then that yes, indeed, there is a significant influx of cryoprotectant in the short protocol. Not as high as in the long protocol, of course, but it's quite significant. And in terms of the osmotic strength within the cells, you can see that in the second step, you reach equilibrium in 3 seconds time. This is amazing. This is not due to the influx of CPA. It's due to the efflux of water. So we get the conditions inside the cells identical to the conditions outside the cells, of which we assume they are sufficient for vitrification. So that means that we can exclude the possibility of intercellular ice formation in those cells. So that's nice. Then we turn together with Florence, we turn to pig embryos. We have done the same kind of work. I started 10 minutes past. Never mind. Pig embryos. We've done the same kind of approach, and of course we find similar results, which is important. Now, we have done also simulations of what would happen if you would have a higher sucrose concentration. Why sucrose? Well, if you increase the sucrose concentration, you can perhaps lower the concentration of the permeant cryoprotectants like DMSO and ethylene glycol, which may be toxic at these very high concentrations. So people have considered the use of sucrose quite often. Here you see what will happen if you don't use sucrose or you use a lot of sucrose. And it doesn't look too good, because if you see here what happens to the volume, the cells are shrinking really tremendously. They shrink so much, they will have perhaps only 14% of the original water content. So they're heavily dehydrated. And this may not be too healthy for those cells. So this kind of modeling will not tell you whether it's good or not. It will tell you what is going on. And from that you can assume maybe I should do this experiment or that experiment. Here you see for all those concentrations of sucrose that yes, the amount of water, the dehydration of the cells is increasing only. It's horrible. And the extracellular concentration of permeating cryoprotectants is of course going down, but the intracellular cryoprotectant concentration is not even going down. So the original thought, does it help? It doesn't even help for the intercellular concentration of CPA. It does not help. So that's why I would say maybe this is not so good idea. Now to turn to the real empirical work done at INRA. Originally Berthelot had used OPS vitrification already with a short protocol already in 2000, and they got live offspring. And then later a similar method was introduced or used by Cuello in Murcia, and they got very good results. So that's quite nice. So using that technique, we have now also in INRA, they have also used vitrification of porcine embryos, but now they have compared the 3-minute protocol with the 2-minute protocol, so a little bit shorter. And they found that the embryo survival in the first experiments, they found that the survival was approximately equal. One time it was better, other time it was not so, that was equal. So overall it was equal. They also derived or found a number of markers for embryo quality that you can use. And now in a recent experiment, Florence has done vitrification again. And now she found no differential expression of markers in the short or long protocol. And in fact, the long protocol had a better in vitro embryo survival. So overall, we think it is not a good approach to use a very short protocol, even though Berthelot originally had done that. So just to end with some thoughts, in the horse oocytes, we use the same short protocol as the Utrecht group, but in those oocytes, people use relatively high CPA concentrations, and they use a very short protocol. But we have used the cryotop instead of the open pooled straws, and therefore we had, maybe therefore, I don't know, we had success. And we had success thanks to, or despite, using the short protocol. But we had no success with the long protocol. But that comparison was not even valid because it was done at a lower CPA concentration. So the jury is still out there. And with the pig embryos, we see that they use a relatively low CPA concentration, which can give reasonable or good results with OPS and super fine OPS, open pool straw. We find now no advantage of the short protocol. And quail in Murcia, they used the 3-minute approach, which gave good results. And the people at the Nagashima's group in Japan, they even used 6 minutes incubation with excellent results. So, and what is the difference? The people in Japan, they used a device that gives even higher warming rate and cooling rates. So I would say that probably the cooling rates and warming rates are very important, and we could perhaps investigate the use of a minimum volume approach that would give these high cooling rates and warming rates, such as was done by the Nagashima group. And with I want to end this presentation and thank you for your attention.
Okay, I'm going to present our work using poultry stem cells for biobanking of avian genetic resources. And I'm Mike McGrew, I'm just introducing this topic. And then Bence Lazar from NBGK in Hungary will present his results, and followed by Marina, who is at INRAE in France, will present her results. The problem we're trying to address is that cryopreservation is very challenging in avian species, and that's due to the structure of the egg or the oocyte, which contains this large amount of yolk, which makes it yolk or lipid, which makes it impossible to freeze. So what we're trying to develop is the use of chicken primordial germ cells or primitive germ cells from the embryo to biobank breeds of chickens. These cells are the self-renewing stem cells that their only purpose is to make sperm or eggs in the adult animal. These cells are diploid, so they can proliferate. And what's unusual for vertebrate species is that we actually learned how to culture these cells in vitro, and they retain germline competence. So this is our whole procedure here. We start with a chicken embryo at a very early stage of development, before it's even formed a nervous system. There's about 100 primordial germ cells in this embryo. We sample them. We could freeze them at that point, but because there's so few, we put them in culture, and they will proliferate and divide once every day. So within 3 to 4 weeks, we'll have almost 1 million of these cells. At this point, we can cryopreserve them and store them in our biobank. At some later point, we thaw these cells, we inject it back into an embryo, which is the same stage. This is now our surrogate host embryo. We hatch this embryo, and then we breed these chickens, and some of the offspring will come from our frozen cell. So now we'll move on to Bence, who will present his results for the biobanking.
Thank you. So today I'd like to show you really shortly our results on the indigenous Hungarian breed, how we use these techniques Mike mentioned to make a gene bank based on primordial germ cells. These are the 6 breeds, the Hungarian indigenous breeds we used, the speckled and the black Transylvanian naked neck, the partridge color Hungarian, the yellow, the white, and the speckled Hungarian. We established in the individual cell lines at least 20 from each breed, and then we made 6 parallel cryotubes for each individual cell lines, and then we store these parallels into different geographical locations just to improve safety. And I'd like to give you an example, the partridge color Hungarian. So we started with establishing cell lines, 21 to be exact, for females and 17 male cell lines. After that, we did a validation step. We used immunohistochemistry with germ cell specific antibodies and also qPCR with stem cell and germ cell specific markers, just to be sure that our cultures or our cells are good quality and truly primordial germ cells. After that, we did an in vivo migration test just to make sure that these primordial germ cells are able to migrate into the developing gonad. So it was kind of a functionality test. And after that, we choose a recipient breed for the partridge color. It was the black Transylvanian naked neck, because the naked neck trait is dominant. So when we did the back crossing with the donor breed, it's easy to select the hatchlings and decide which one is donor derived. So we used cells from our established cryobank and selected a male and female cell lines. We did the injection, as Mike mentioned. In 5 trials, we injected 52 embryos, and then we got 24 potentially chimeric black Transylvanian naked neck chickens, 13 roosters, and 11 hens. And after that, we did the back crossing with the donor breed, and at the end of the process, we got 14 donor-derived hatchlings, which is around 4%. And we could prove in 4 cases that these black Transylvanian naked necks are truly germline chimeras, which is around 16%. And that was me. Thank you.
So, since the amplification in vitro of PGC and the cryopreservation prior their use for the reproduction, the PGC are amplified in vitro and cryopreserved prior reproduction, it is important to know whether these steps affect their integrity. So we addressed this question, and we studied the impact of long-term 6-month and 9-month in vitro culture compared to 1-month in vitro culture on the gene expression in our primordial germ cells, and also on DNA methylation and germline transmission. To study gene expression and DNA methylation, we used RNA sequencing and reduced representative bisulfite sequencing approaches. So the main finding of our RNA-seq study was that long-term culture had a strong effect on the gene expression in male and female PGC. And this effect was more important in the case of male PGC. In contrast, cryopreservation had no effect on gene expression. As revealed by RRBS study, this change in gene expression during long-term amplification of PGC in vitro was accompanied by the change in DNA methylation. And on this graph, you can see that the percentage of CpG methylation significantly increase in 9 months male and female cultures. In contrast, in transcriptomic analysis, cryo preservation had the impact on DNA methylation in the same manner for male and female PGC. Finally, we studied germline transmission of our PGC culture, and for this we injected male and female Noir de Berry long-term and short-term cultures in Rhode Island host embryos, and obtained by this way chimeric germline chimera, male and female birds were crossed with Rhode Island hens and roosters, and in combination Noir de Berry Rhode Island, the black color is of Noir de Berry is strongly dominant, and this allows to screen easily the origin of other progeny. So here we can see the first French progeny from cryopreserved derived primordial germ cells, and we obtain it for some chimera birds, for male and female PGC, very good transmission rate, you can see in red in the table, and moreover, we could not obtain the progeny from long-term male PGC cultures. In contrast, we obtained progeny from long-term female PGC cultures. So this may indicate that the impact of long-term cultures, which was more important for male PGC, compromise the reproductive capacities of these cells. Overall, these results may contribute to better master PGC cultures, and also to recommendations on PGC protocols and utilization in the FAO guidelines. Thank you for your attention.
But you can also use a gonadal for cryopreservation and make collections like that. Krisztina.
Okay, so nowadays, okay, sorry. So nowadays, sperm freezing is the only practically used preservation method in avian species. Cryopreservation of the oocytes or embryos is almost impossible because of their biophysical traits. And as the female are heterogametic and the males are homogametic, the female genome falls out of the gene conservation. In the image project, the task of the Hungarian team was to elaborate alternative methods, how we can keep the female genome. We have two ideas. One of them was applying gonadal tissue transfer, and the second one to create interspecies hybrids as a potential surrogate host of PGCs. At first, I would like to say some words about gonadal tissue transfer. Although it is impossible to cryopreserve the egg, but it is possible to freeze the ovary of the day-old chicks because its construction in this age is different from the adult one. So if we graft the frozen tuft gonadal tissue of one-day-old recipient chick into one-day-old donor chick into one-day-old recipient chicks, and after sexual maturation, we inseminate this gonadal chimera hens with the frozen tuft semen of the donor breed, we can obtain the donor genotype in the F1 generation, so in the first progeny generation. We improved the effective vitrification method of gonadal tissue. Acupunctural needle was used as a gonad holder, and we store them in liquid nitrogen. We improved thawing method too. What is important that you can cut the gonads into small pieces, and it's enough to transplant a little gonadal tissue piece because it will be a complete gonad after sexual maturation. It was proved that not all breeds are suitable recipients. Information obtained from evaluating genetic differences of intensively selected lines in which there was successful pairing was compared in the indigenous breeds. According to the results, donor-recipient combinations were created which could be effectively used for gonadal tissue allotransplantations. We found that white leghorn and Novogene white are good recipients for yellow, partridge color, and speckled Hungarian, as well as blackered and speckled Transylvanian naked neck indigenous chicken breeds. We obtained donor-derived progeny from frozen tough chicken ovarian tissue, which was not published before. 21.5% of the laid eggs proved to be donor-derived, and the remaining one were recipient-derived. So we were thinking whether a sterile hybrid would be a good general recipient for gonadal tissue transfer too. We published our results in Animal Reproduction Science. And parallel, we worked on our other idea to create interspecies hybrid as a potential surrogate host for PGCs. Mike McGew and his team in the previous presentations showed the role of the PGCs in the gene conservation. So if we create a sterile recipient, the benefit is that there is no need any treatment against the recipient's own PGCs. And in this case, if we inject the donor PGCs into recipient embryos, we can obtain donor-derived germ cells in 100% from these germline chimeras. So it is the general outline of producing donor-derived hatchlings with sterile recipients. You can find interspecies hybrids in the nature too. For example, when you keep together domestic fowl and guinea fowl. This is a yellow Hungarian indigenous chicken breed, and here you can see their hybrid progeny in different ages. We can improve the number of the hybrid progenies applying artificial insemination. We crossed guinea fowl females with domestic fowl males, and the hatching rate of hybrids was 6.65%. We tried the opposite crossing, applying domestic fowl females and guinea fowl males, but in this case, unfortunately, only one egg hatched out from more than 700 eggs, so it was not really effective. Only the guinea fowl female and domestic fowl male crossings are successful. According to the histological analysis, all hybrids are males and sterile. The gonads of hybrids contain normal tubule structure with inactive germinal epithelium. The hybrid status was verified applying microsatellite marker analysis too. In order to test the functional and structural integrity of the hybrid gonads, 147 recipient embryos were injected with GFP-expressing PG cells in 3-day-old embryonic life. And this type of cells help us to follow them in the developing gonads. 85%, we opened the injected embryo in the 7.5, 14.5, and 18.5 embryonic age, and 85% of gonads contained GFP-positive cells. Here you can see the PGCs in the developing gonads of a 7.5-day-old embryo. And here you can see the PGCs in the gonad of 18.5-day-old embryo. So crossing guinea fowl with domestic fowl was successful, but only with female guinea fowl crossed with male domestic fowl. The observed offspring from the successful crossing were sterile male hybrids. These sterile hybrids have endogenous germ cells, but they don't develop into sperm cells. The sterile males produced in this study might be suitable recipients for male chicken PGCs and possibly for female PGCs. We published our results in the scientific report, and this year we plan to inject of indigenous chicken PG cells into hybrid recipient embryos to investigate of germline transmission, as well as we would like to create new hybrid constructions using pheasant and chicken, because it was published that there are females among their progeny. And we would like to use this sterile hybrid for gonadal tissue transfer experiments. Thank you for your attention in the name of my colleagues who took part in these investigations.
So thank you for your attention. Maybe one question. You've seen there was some work on gonads, but also you looked at also PGCs for the recipient, hybrid recipients. Maybe clear for everybody.
Okay.
You know, the PGC are in the gonad, you know, after a while. But not first.
Okay.
No question?
Okay.
Okay. So you see different stages. And we follow the stage of differentiation of the germinal cells. And the last talk will be on the differentiating cell, germ cells, that is the gametes, and of course the more usually common gametes used for cryobanking is semen. And so Julian will speak to you about that. Don't forget semen, that is also very useful for cryobanking.
Thank you, Elisabeth. Now I go to try to make a general overview about the sperm cryopreservation for establishment of the sperm cryobank. It's important to stress that it is estimated that the 30% of animal species, including mammal, amphibians, and birds, will be endangered, will be extinct in the next years. Currently, around 1,300 species of threatened birds in the world are endangered. The main cause of threat is climate change, intensive farming, poaching, pollution, inbreeding, genetic hybridization, etc. And regarding the fowl, the gallus species, for example, the wild fowl, the Indian red jungle fowl, or the Sri Lanka jungle fowl, the first cause of threats is the habitat destruction, the poaching, the breeding, and the genetic hybridization. The half of the domestic poultry breeds are currently considered to be endangered. And the mass of them, the Gallus gallus domesticus, are native chicken of breeds. The main cause of threat of the Gallus gallus domesticus are the intensive farming and commercial needs and the restructuring of rural areas. So in this context, it's very important to the establishment of the sperm collection and sperm cryopreservation. One ancestry species can have a scientific use as an animal model on health and reproduction research activities. For example, the Indian red jungle fowl show a great resistance to the glycerol as a contraceptive effect. And we think that this maybe is a useful model for to study the cryoprotectant effect on sperm and chicken and sperm storage tubulus. Threatened breeds can have used as a basic biological research into physiology, diet, reproduction, or climate tolerance at the physiological and genetic level. The native breeds with unique characteristic of tribe could be also helped with identification of sperm genes involved in natural disease, parasite control, thermal stress, and of course, purlines with unique product traits are very useful for integration of the valuable genetic material into different production system through artificial insemination. So, we have, my colleagues have talking about the PGCs, cryopreservation, other alternative is cryopreservation of gonad, but the sperm is the non-invasive and the more is not expensive regarding with the other methods. And the sperm banks allow to have a precious germplasms for a long period of time and to restore genetic diversity by main artificial insemination. Is the more feasible alternative. I would like right now to indicate the scientific milestones in sperm cryopreservation. Unfortunately, it's my opinion, we have not advanced very much in the last 50 years. In 1940s, Schaffner makes the first attempts to freeze roasted sperm. In 1949, Poultry discovered the cryoprotective action of glycerol. The glycerol is the first spermian cryoprotectant in all the species, in mammals, in fish, etc. Was discovered this effect in chicken sperm. In 1955, Allen confirmed the findings about the contraceptive effect of glycerol after artificial insemination. Poultry also see this effect of contraceptive action after artificial insemination. Lake designed a glutamate-based extender that currently we have used in the majority of laboratories of spermatology of roosters, based in the composition of the seminal plasma of roosters. In 1968, Lake used liquid nitrogen for successfully freezing chicken sperm, and in 1977, with Stewart, he proved this freezing protocol with a cooling rate. The lowest temperature is lower until minus 35 degrees Celsius, before semen was put into the liquid nitrogen. In 1999, a Russian group of Selutin studied the successful alternative to cryoprotectants of glycerol and the methods. You use the DMA as a permanent cryoprotectant in pellets. In 2005, Blesbois provided new insights about the role of sperm membrane fluidity and cholesterol content on the ability of survive cryopreservation. And in 2006, Long provided new insights about the sperm bioenergetic and freezability. It's a brief summary about the first, in my opinion, scientific milestones. Yes. We have an example for the FAO and the Initiative for Domestic Animal Diversity recommendation for a cryobank in an endangered small population. They indicate that a total number in these guidelines of 6,544, with a minimum of 20 males, with proven fertility and proven sperm freezability. So the number of doses per male is 327. Consider that 5 doses is get per sample. We need about 5 months for creation of a cryobank in a small population. Well, it's not realistic for most native breeds, unless for the Spanish breeds, because we usually get only 2 doses per sample. So we need about 164 samples per rosters, and we need more than 1 year for the creation of a cryobank. We have made a cryobank with different Spanish chicken breeds. In 2009, because the risks of flu in Spain, the Spanish National Institute for Agriculture and Food Research, is by INIA, my institution, supported by the Spanish Ministry of Science, supported the establishment of a germplasm bank with 12 Frasenet breeds. It's the first cryobank of chicken in Spain. It's a very dynamic cryobank because there are a constant update of cryopreserved dose using the latest cryobiological techniques. It's an excellent complement for ex situ in vivo conservation program in the avian experimental unit from INEA in Alencin. And Morbo is very interesting because we use these breeds as a model for research activities in genetic, in reproduction, to looking for new animal welfare markets, to study the different management, free range management, etc., etc. Which is the limits of the implementation of a chicken sperm cryobank. But the first limit is the high variability in the result of the fertility after using sperm frozen thawed. It is mainly due because the high susceptibility to cryo damage during freezing thawing process, which is the traits of a roasted sperm determining this high susceptibility to the freezing thawing process, which is the filiform. It's a filiform cell with a very long flagellum, show a low sperm osmotolerance, is very sensitive to oxidative stress, rapid induction of a chromosome reaction in vitro, in the laboratory, and the susceptibility of DNA to the cryo-injury. Sperm not contain cis-chain residues and thus lacks the potential stability in the effect of the sulfide bonds of mammalian sperm chromatin. In addition, there are an endogenous factor, an environmental factor that is influencing in this sensitivity of sperm to freezing thawing process. For example, endogenous factor, the breed and the individual, the seminal plasma, and within the environmental factor, the photoperiod, the temperature, the social interaction. This is very important in free-range condition, the most of breeds of chicken in Spain is maintained in free-range condition. Okay. Other factors, the diet and housing system, and the subclinical health problems. For example, the presence of ectoparasites. Some breeds show best freezing thawing response than other ones, 2 minutes. Oh. The presence of seminal plasma may affect the response regarding to DNA fragmentation damage. I am going to very, very fast. Decision is important in free-range condition. The motility variables, the total motility, the straight line velocity, curvilinear velocity of sperm are higher when the samples were collected in the spring and unfreezing in the spring. The social interaction are very, very interesting. For example, in males, in the presence of females, the motility variables are higher than in that roster isolated from females. For example, straight line velocity is about 40 micrometers per second, and in males, and the same males is isolated of hens is decreased to 30 micrometers per second. It's also important to access to pasture, for example. Outdoor horses system with access to pasture improve the motility in fresh semen. In some breeds of chickens, for example, in red barred basca, we obtain better results after freezing thawing process. Subclinical health problems, health factors are very important in free range. For example, the presence of parasites in some animals to induce a stress for heating and skin irritation that may affect the cryosensitivity of sperm during process. Now, together to this low reproductive success, my colleagues have indicated this problem. Only the male genome sex chromosome is conserved because bird females is the heterogametic sex. So it is the second first limitation. And when through the image, we will have to try to develop a new combination of cryoprotectants to improve, to optimize the freezing thawing process, and the other alternatives for PGCs, cryopreservation, and tissue gonad preservation have been indicated by my colleagues in the last speech. Yes? Okay, on time. Thank you for your attention.
So thank you, Julian. So it is the end of the session. Well, yeah. Now we can have some time for discussion. The discussion maybe that could be on around which, what about all those different cell types were there for use for cryobanking, for collection, for reproductive collection in cryobank. I don't know if it is clear for everybody that all these sorts of tissues or cells, in fact, have their place in the collection. So, I don't know if you have some question about it. Good question. Oh. Yeah.
No, I think it's not a specific question, but then since we have seen you have diversified the choice of technologies, but then now as a recommendation for the gene banks or the farmers, so maybe we should have sort of a decision tree in which condition you take this one or that one, so that they know exactly what they can choose corresponding to what they expect. Also, I was a bit surprised that you can say that for one breed it can take, I did not understand, but this is we can discuss at lunch, why it could take so many months to sample a local breed, because well, what we did that we moved in France. We moved the males to a single place, and so we can collect enough for local breed in 2 months, which is already long. But generally, it's more the frame because so that the gene banks are not lost in all the different options. How they can choose, and including the success rate, what they can promise to their users. Because that's always the main issue. If we keep something, that's because we expect we can use them. And if it is a 4% success, but in males, when you have 5 males in cryobank and only 1 male has progeny, well, it's 20%, but it's only 1 male. So really being able to have all the elements in hands so that they take the best decision they can.
It's a very good point of discussion. We will not have the solution. Probably these 3 decisions, it's not so easy to make since the discussion we started to have yesterday between us indicated that depending on the country, you have not the same infrastructure, and in fact, the tree will be different. If we take the example and also it depends on the breed you have, on the number well, probably on the facilities that you can get to have technical level of technical schedule. It's not so easy. So these 3 of decision we could make a theoretical one, sure, but knowing that it will probably still be theoretical, since after it will have to be adapted to each situation. And probably one of our recommendation would be to, if possible, not put everything in only one method, in only one collection. Try to have a diversity of collection, diversity of type of cell in the collection, if possible, since in fact each of them have not the same return, but not the same also capacity of use, depending. You don't know when you will use the collection at the end. You do not know. So you don't know if at the moment when you will use this collection, you will get the technical skills useful for this collection or for the other. So probably, but we did not yet discuss really about that, probably we will suggest try to get 2 different type of cell in a collection for 1 breed, if possible. Because in the future, you do not know how will be the condition that in which they will be used. You agree with that?
Short-term and long-term approach, and whether you want to use it often or not, depends very much.
So two things before lunch. We have lunch now. It is at the cafeteria of the faculty. Those of you who have no lunch ticket, please take it just at the outside of the exit of this conference room. We will be back in a big hour, but we should start at 13:15. And I wanted also to tell you another thing. The facility where the genotyping has been done for the IMAGE project is represented here by Frédéric Antigny, who has a poster just in the front of the entrance. And if you are interested, he can tell you and answer questions now at lunchtime or later in the coffee break. So just that you know this issue. So go and have lunch, and please ask for the tickets if you don't have them. You need a ticket for lunch. You can leave all the computers here. We can lock the doors. So this is possible.